Hi guys, welcome to In Vivo. You need to be able to explain the importance of sticky ends, explain how a DNA fragment can be inserted into a vector, how then the vector can be introduced into host cells, and describe the nature of gene markers and explain how they work. So we're looking at this part of specification, and last time we were looking at the isolation. Today we will be looking at insertion, transformation, and identification. So let's get started with the importance of the sticky ends that we've seen in the process of isolation. So those were our sticky ends. As we can see, they live on per basis, which then can be used to be stick with another part of DNA from the other source. So we can do that by cutting the other bit of DNA using the same restriction endonuclease. So by doing this, we are going to get the complementary sticky ends. So preparation, so how can we prepare the DNA fragment for the insertion? So we need extra length of DNA. We also need the RNA polymerase. We need a promoter, so it's a binding site for RNA polymerase of the region of DNA. We need nucleotide bases of promoter that attach both RNA polymerase and transcription factors, so transcription can take place. And we need a terminator, so that's the region of DNA that releases RNA polymerase and transcription. Okay, so all those processes are needed so the protein can be transcribed. So how we can finally insert this DNA into a vector? Okay, firstly, what is a vector? Vector is a plasmid. So it's a single loop of DNA found in the bacterial cell. The job of it is to transfer, to carry foreign DNA into bacterial cell. Okay, could be any cell, but the idea of the fact that you need to say cell, it's important, it's on the line. Okay, so this was our DNA that we've isolated using a restriction endonuclease. We're going to use the same restriction endonuclease to cut the plasmid, so the complementary sticky ends will be produced on both of the sources of DNA, and we can insert our gene of interest into this plasmid, producing recombinant DNA. So how can we join them together? We can do it by using enzyme called DNA ligase. So DNA ligase, it's an enzyme that will join those pieces of sections of DNA and produce the bond. Right, so how does it work? So remember, we had a DNA of our own interest. That was a double helix. We've used the restriction endonuclease to cut this DNA and produce the sticky ends. Now this is the other source of DNA, the source from the vector that comes along, and we can cut this DNA using the same restriction endonucleases. So then we would like to join those two fragments of DNA together. We can do so by using DNA ligase, so the double-stranded DNA will be produced. Okay, so this is how it looks like when you will put it into the plasmid. Okay, so this is a plasmid incorporated with the new DNA. Okay, so step number 3 is the transformation. So we did isolation, insertion, now time for transformation. So transformation is the introduction of this recombinant plasmid that contains our DNA of our interest into a host cell. So of course, host cell, bacterial cell, it has a cell wall which is not permeable to the plasmid. So we need to make it permeable. How we can do it? We can do it by growing those mixing plasmids with the cells in the medium containing calcium ions and those calcium ions, together with the changes in temperature, can make this bacterial cell wall permeable, so the plasmid can pass through the cell surface membrane. Okay, so growing them, they can pass on. Right. Finally, identification. So we need to now check if this host cell has taken up the plasmid. So we can do it by three ways. So we can use resistance to antibiotic, we can use fluorescent proteins, and we can use enzymes. So have a look at the antibiotic resistance first. What do we need to remember? It's the fact that plasmid can carry at least 2 genes for resistance to antibiotics. So how we can then check if the bacteria has taken up this plasmid? We can remove the one of the genes. We can cut this location here to insert our gene of interest. So by doing so, this plasmid, this bacteria carrying this plasmid, is not going to be able to carry the antibiotic resistance to tetracycline anymore. Okay, so we can now use this information. So how we can use it? So we can identify the growth, okay, of those cultures containing the tetracycline. So that if they growing, that means that they didn't take up our plasmid. But if they don't, that means that the cell will be destroyed and that cell contain our gene of interest. Okay, so let's have a look how this can be done. So the bacteria is grown on the ampicillin. We haven't used it. Okay, we haven't used this antibiotic to produce this gene for the antibiotic to insert our gene of interest. So of course they should be growing. But what will happen if we will treat them with tetracycline? Of course, our colonies will miss, and those missing colonies tell us that that bacteria, okay, have taken up the plasmid. Okay, final one, fluorescent markers. So those are lovely fluorescent proteins that can just glow. So where they coming from? They coming from the jellyfish because they produce the green fluorescent protein. And if the bacteria has taken up the plasmid, they will just glow. Okay, this process is it's much faster than replica plating. It's faster than using antibiotic resistance. Okay, and the final way is to use the enzyme markers. So they uses the gene that produces the enzyme lactase, and lactase will turn a colorless substrate blue if it will act upon this recombinant plasmid. So this required gene is transplanted into the gene that makes lactase, and if plasmid is present with required gene in the bacterial cell, the colonies will not produce lactase. Okay, so there won't be any color change. So any colorless spots will identify our recombinant plasmid, so the cell that carries that gene of our interest. Right? So that's everything for the another processes. So we've got isolation, insertion, transformation, and identification out of the way. See you later.